sorry. To connect the dot again, what I was trying to say that we have enough number of diseases on this planet to work on the choice of your disease. And you all being the faculty across the country, I don't know you're participating from within the state or different state, doesn't matter. We have to really think pragmatically to see how we can contribute to this very huge gap. There are no treatments to so many diseases. So the onus is on us as a scientific community. And therefore,
Manoj,
we as a scientist.
Manoj,
Yes,
sorry to interrupt, but can you please put it on the slideshow mode, your presentation? Because the participants are putting the message that the slides are not visible to them. Clearly visible.
Yeah.
Is it okay now?
No.
Earlier it was okay.
We are seeing the 2 slides on the screen.
Oh,
So you are the next slide. Yeah. So PowerPoint mode or some slideshow mode.
It is already on the slideshow mode.
Okay.
Wait. But it wouldn't serve the purpose because I have 2 screens connected to my PC. That's why it might be showing you 2 screens. They cannot read it. Or, yes.
Is it possible to close the right one, the smaller one?
No, that's, I don't think so. That would be a possibility. Next.
The previous one was better, quite better.
Wait. Now, is this okay?
Yeah.
Yes.
You can also minimize the lower side of that slides.
Right.
Left side you can reduce one. No, sir.
What do you want to do? Tell me.
The previous one was better.
Okay, good. Life is.
Yes,
I think this is better.
It will work. Please, sir, please.
Yeah, we will make it work somehow.
Yeah,
because I have two screens attached. Now I cannot detach those for this.
No issue, Manoj. We can go with the previous one, which you had shared earlier to this view.
Is this okay?
No, the earlier one, like where we were able to see the slides also on the left hand side.
Now, is it okay or no?
No, sir.
No, sir.
Wait, let me stop share. Wait a minute.
Yes, that can be done. Actually, एक स्क्रीन शेअर केले तरी होऊन जाईल असं वाटतं मला, दोन्ही स्क्रीन शेअर करण्याच्या ऐवजी.
हो, पण मग आता
Otherwise, you can share your presentation to the co-host. Otherwise, it will also work.
Now, is this okay? पण नाही ना, तुम्हाला ते दोन. You are seeing 2 screens. They cannot see anything or they can see something? That's my next question.
Something they can see, but it's not readable to them.
Okay.
No issue. You just share the screen now, Manoj, sir.
Yes.
Yeah.
Now check. Can you see?
Yeah, it's loading. Yeah.
Yeah, leave it there.
This is fine, sir. This way only. Don't go into the slideshow mode or that.
Yes.
Yeah.
If I do that, then it becomes what I will do. I'll increase the zoom, right?
Yeah.
Now, is it? check with the other members.
It's fine, sir. It's fine, sir, now.
This must be better now.
Yeah, yeah. Yes, fine, fine, Manoj. You can just continue. Yeah.
Life is challenging in the electronic
Yes, yes.
age.
Please continue. Yeah. Thank you, sir.
But don't worry, my fellow scientist, listen to me carefully. That is more important. Means, of course, I know you would want to see the slides, but here is a technical glitch. But we will try to, and I will try to be as simplistically explaining to you so that you can get what I'm trying to say. See, okay, so let us start again. Okay, so I was at this slide saying that there are too many diseases where we do not have treatment options available, and while doing so, it is very important as a scientific community that we can focus on connecting the dots, or doing an efficient drug development. When I say so, what I mean that I am a strong believer of a drug development through translational approach. What does that mean? It is in simple words what I can say. We should be able to convert the observations into interventions. What I mean by that, when we are working in a laboratory or early drug development stages from discovery, if you see on the right hand side of the screen or the cartoon in this slide, we start any drug development program with a basic research. Once we have crossed the key steps in the basic research, especially discovery phase, we tend to go and evaluate the interventions in the appropriate preclinical setting, meaning we will evaluate those molecules or leads in appropriate in vitro and in vivo systems. Imagine you have crossed that stage. Eventual and sequential step of the drug development goes to the clinical development, where you will study that intervention which you are trying to innovate, or which is under the development, into a clinical setting. You will give it first time into the humans, followed by, if you have got desirable effects, means safety is established, then you will slowly escalate into efficacy, which is nothing but where you will see whether your intervention or drug under consideration is effective in target disease condition in the target population. Once you have crossed all the clinical development phases, you as a sponsor or a developer would submit that observations which you have done in the lab and in the clinical setting to the appropriate regulatory authorities. Once you get the appropriate regulatory approval, eventually the drug or vaccines which gets rolled out into clinical phase, in a clinical implementation phase, and eventually, as you know, it will go to the public at large. So what I am trying to say that we as a scientist should be able to convert the observations, and I strongly believe in a phrase called as discovery to recovery. Means, as you started your discovery process right from here, now you have been able to cross all the hurdles or stages of drug development, and eventually your drug has reached to the public. And when I mean to say that it has reached to the public, meaning it is being able to use for the recovery purposes, or treating or diagnosis or prevention of any disease purposes for which you had started that process of drug development. So you should be, or we as a scientist should be able to connect the dots, go backward from the field, go back into the lab, and find out the shortcomings of each drug, because no drug is devoid of any limitations. Each drug on this planet has its strengths and weaknesses. So we should be able to, very methodically should be able to identify the current limitations of the drug during the process of drug development, or even after the drug is approved and is being used in the public setting. So you all are aware that, drug development is not it's not a simple process. It is a very complex process which involves a lot of resources, expertise, and coordination of experts like you and me to develop a single drug. And the cost which is involved in current scenario to develop a new drug, brand new drug from scratch, is unmanageable from any country's perspective. $2 to $3 billion to develop one new drug from scratch. So it's very time intense, resource intense, and we need significant expertise to come together to develop a meaningful drug going forward. So while I have made that statement, why it is, you must have been reading that the drug development pipelines are drying, more rapidly than it was in the past, because the attrition rate of new drugs is significantly high, and you are studying in your own labs how drugs are getting killed, or they are out of the context because of lack of appropriate methods being followed and appropriate logics being applied, and therefore significant high attrition rates are being observed. And as I said, 10 to 12 years to bring one drug, a brand new drug from scratch to the market, it's a long time. And while doing so, the regulatory requirements are changing day by day. You would agree with me, 20 years ago, the requirement of registering a drug was entirely different, be it India, US, or anywhere in the world, versus in current scenario. Every country and their regulatory bodies are coming up with more stringent rules and regulations and making the landscape of drug development even more complicated. But that complication is meaningful. It is not just for the sake of doing it, but the real context of making it more complicated or streamlined is to ensure that the drugs which we are developing, those drugs has to be safe and effective. That is the real context. And therefore, as I said in my previous slide, that there is an unmet medical need to develop new drugs for lifestyle diseases, more severe in the infectious diseases. And the classical example, we all have gone through, or we are still going through, the pandemic. And we know by now the whole world was very conveniently ignoring the infectious diseases domain for drug development. And now post-COVID, I would say it is not, as a post-pandemic, hopefully the bigger organizations globally will try to focus on treating infectious diseases. And why I'm saying so, antimicrobial resistance has taken a center stage now, and bigger organizations like WHO, DNDi, and all regulatory bodies across the globe are concerned and alarmed to develop drugs for infectious diseases. And that is where, when I will continue, that we as a scientific community, especially from Indian perspective, can take the lead in this specific domain of infectious diseases drug development. This next slide gives us an idea that, what are the different stages of drug development. Let us start with some basic research. In that context, we must know what is the target disease, what is the target pathogen. Once you know this target disease and target pathogen, then target population should be known to you. Once you have this basic understanding, then as a scientist, we can go to the molecular and pathophysiological details about that disease in context. To give an example, COVID, 3 years ago, none of us on this planet were aware of COVID as a disease. What we knew at that time, only an unknown virus has been spreading globally, causing havoc and killing people left, right, and center. So as we were, unknown to the pathogen, at the same time, the pathophysiological changes which were happening within the body, the scientific community across the world, ramp up their efforts to understand the disease first. What was the virus? Then how virus is infecting and how pathophysiological changes and pharmacological changes are happening within our body, leading to a condition called as COVID-19. So that core understanding of the disease is very important. While that is happening, early discovery means what are the screening and how screening is done, how hit molecules are identified, which will lead to a more appropriate lead molecule with desired physicochemical and biological properties. Once you as a scientist have crossed this phase, eventually you will assess those lead molecules in appropriate in vitro and in vivo animal models, especially doing MIC or effectiveness assay in appropriate cell lines, followed by safety and efficacy studies in appropriate animal model, be it rodent or non-rodent. So once you have completed this phase of, I call it as an early or preclinical drug development, then the next stage of any drug development program is opening an IND. Means IND is nothing but investigational new drug application. So once you have crossed these stages of early drug development with sufficient safety and toxicity as well as efficacy data in hand, you as a scientist or as a sponsor can go to the appropriate regulatory authority and open an IND application. Once that IND application is opened, based on the safety and efficacy data of your molecule in hand, an appropriate CMC consideration, which is chemistry, manufacturing, and controls, that is, in simple words, formulation characteristics and its stability and other characteristics, needs to be assessed and addressed so that you have a robust plan in hand to be able to go to first in human stage of drug development. While that is going to happen, it is again, you all know as a scientist and faculty that it can be divided into phase 3, 3 phases, starting from phase 1 to phase 3. There are several studies we have to conduct to be able to prove, again, 2 things always: safety and efficacy of your molecule in hand. If that is proved in the reproducible manner, you as a sponsor have an ability to file a new drug application with all the information which you have gathered over last 8 to 10 years and spent countless number of money, with a very cross-functional interaction among the scientists across the your different divisions in your organization. So then once you have submitted the NDA, the appropriate regulatory authority have an obligation to review the data submitted in appropriate formats. Once the agency is assured of the safety of the drug, always remember, I'm talking about safety more than the efficacy here. Each regulatory authority, let it be DCGI, CDSCO, FDA, or European authority or Australian regulatory authority, the paramount focus is always on the safety. If the drug is safe and efficacious, then the regulators are comfortable to give approval to appropriate interventions which is under consideration. So I think this slide gives us a very good understanding that how the drug development process works and moves across the different stages. So while doing so, I just want to walk you through this very basic fundamental explanation why we have to develop drugs. And the slide itself is self-explanatory. As you and I are healthy as we speak today, everything is happy and wonderful, right? Look at that small kid. He's happy, he's smiling so happily. But the moment there is something abnormal has happened in our life or within our body, it leads to a condition called as disease. When I say abnormal, let us take a simple example. There is an increase in the blood pressure, say hypertension. So what you and I as a scientific community, or let us take the example of COVID-19, which we all can relate very quickly. What happened in COVID-19? I got an infection with SARS-CoV-2 that led me to decrease in the immunity leading to clinical signs and symptoms like cough, cold, and if not treated, and if I have comorbid conditions, ARDS would happen and the patient will go into a different critical situation. So something abnormal has happened within our body. We as a scientific community across the globe, be it any pharmaceutical company, any biotech company, or a vaccine developer, are just trying to bring normalcy within our body. Period. Only where the whole scientific community, pharma community is just trying to restore this normalcy, trying to restore the normalcy from this abnormal state, which has caused due to an intervention, which could be a microbial or lifestyle disease condition or a rare condition. This principle can be applied to any disease condition. You name it, only two things are happening. Something is missing or something is in excess. And we are trying to restore that abnormal equilibrium which is lost to the normalcy. So remember, this slide is very important because it has a connection to the next slide. And as I said in my previous slide, that we as a scientist have to be always focused on. If you understand this slide which I am showing, believe me, I know you all are learned faculty and senior faculties joining to this interaction. If we understand this slide, we can teach our students a good way of doing research and drug development. Always, what my firm belief is that any drug, be it a drug or a vaccine, I mean to say drug or biological, it has to be safe. The safety of that intervention is paramount. Not only safety, while we are talking about the safety, the efficacy of that intervention. I will say drug for ease of communication, the drug has to be both safe and efficacious. Now, when I talk about safety and efficacy, how do I will evaluate that, or how do I know if my drug is efficacious? So at the same time, we as a scientist, or you as a drug developer, should be able to know what is your target disease. Let us take an example of COVID-19. So COVID is suppose you are developing a new drug from phytopharmaceutical origin and your target disease is say COVID-19. Then what would be your target population? Now COVID-19 is a drug for the whole globe. It doesn't spare the pediatric to geriatric population. So that becomes your target population. So when you have identified your target disease, say COVID-19, target population is the whole population on this planet. Now, while you are developing your drug, the paramount consideration has to be on your safety component. Whether your drug is safe, if the answer is yes, ask next question, is the drug which you are developing is efficacious? If these two things are satisfied, at the same time, when you will submit, as I explained it to you in the previous slide, that when you have completed the whole drug development process and submitted the application for approval to the appropriate regulatory authority, the paramount responsibility of these regulators is to see whether the drug has a benefit to risk ratio is high, meaning whether your drug is bringing more benefit to the patient as against the risk. And while all these things are happening, what we as a scientist are doing, as you move across the spectrum of drug development, if you remember my previous slide, from preclinical to clinical to NDA submission, what we are doing as a scientist, we are learning in each stage of drug development. Simple, you conducted an in vitro study, you have determined MIC, minimum inhibitory concentration, or IC50 of your drug. So you have learned, right? So you will repeat the experiment, say n is equal to 3 times. What you are doing, you are just confirming. So you are learning and confirming. So you are again going back to the next experiment. A preclinical animal efficacy model, say antifungal drug. What you are going to do, you will run that experiment. You will learn that the drug is effective at 3 dose points, 1 milligram, 5 milligram, and 10 milligram. So you have learned, but you will try to confirm it, showing that whether which dose is more effective, going back to this square. While the drug is found to be effective, at what dose, up to what maximum dose the drug is going to be safe, right? This is an interplay between only two things. Across the spectrum, across the world, we are always, every scientific community, every pharma company, every biotech company, every faculty like you must be and always thinks about only these two things. Is your drug efficacious and safe? If that is happening, we are on the right track. And at the end of the day, the benefit risk ratio is paramount. Now I'll give you again one simple example. As far as the COVID is concerned, several drugs were given an emergency use authorization. That means based on the initial preliminary clinical evidence, the agencies across the world assess the benefit risk ratio, whether the benefit of giving this drug is higher than risking the patient, means causing adverse events. So based on that benefit risk ratio, which was favored in the positive side, saying that it is showing more benefit than as against more people dying, the EUA were given to expedite the rollout of the drug to be able to prevent the deaths happening from COVID-19. So try to connect the dots what I have been talking about. You should know the target disease, target population. Once you know these two, as you continue your drug development process, your paramount focus should be always on the safety and equally on the efficacy. Because if the drug is too safe and not efficacious, it is of no use. But if the drug is efficacious but it is not safe, again it's a tricky situation for you. So there has to be an appropriate balance as we continue the drug development. Now I am coming with this background. I hope I have set the stage for you guys to understand that what we as a scientific teams have to think through when we are planning our drug development program. Or I know most of you are faculty, meaning you would have your own labs, your students, and you have to identify and give some projects to your student. So my recommendation would be to make them aware about all these things as you assign them some meaningful programs, and those has to be thought through very well, so that they just don't do one more experiment, use the resources at your institution and just finish something and submit a thesis and they are gone, but the work has not moved meaningfully forward the way it should be done in a translational manner or in a team-based fashion. So I believe these factors needs to be very well thought through and conveyed through. And my recommendation is one more, that no one person can do drug development, period. There is no ambiguity that if I am saying that I alone, Manoj alone, can develop a drug, I could be the most foolish person on this planet. So let us be practical and pragmatic that we need different set of experts to develop any drug, be it a natural drug or a modern drug, a biological or a diagnostic kit. We need multiple brains to work at the same time with the same mission and goal. So now I'm coming back to your context, phytopharmaceuticals. Why we are talking about phytopharmaceuticals? Because this particular faculty development program is focusing on phytopharmaceuticals. Now, I don't need to tell you what is the meaning of phytopharmaceutical. It's nothing but something which has come from the natural origin. This is my simplest understanding, which you are trying to standardize and purify and make it available to be able to show its efficacy and safety. Going back to those two words again, everything will revolve around those two words, right? So as far as the phytopharmaceuticals is concerned, why there is so much of traction these days focusing on phytopharmaceuticals? Because globally there is a ever increasing demand of phytopharmaceuticals and herbal extracts. what is happening, including the dietary supplements, and especially US is the hub, or you may not believe, every US person takes at least 2 to 5 dietary supplements, medicines every day, just for the sake of taking, right? They need some multivitamins, they need some herbal tablets and solutions and suspensions and whatnot. So coming back to the context, the global phytomedicine and herbal extract market is exponentially increasing, and it is expected to be more than 15 billion in next 6 years. Meaning there is a significant growth of almost 8% growth is likely to happen in this sector. Now on the right hand side, you can see several companies. These are very key companies across the globe, which are leaders or players focusing on the phytopharmaceuticals. And I was happy to see that some of the companies from India are among the top companies in this space of drug development. Not drug development per se, I would say, in the space of herbal extract manufacturing and selling it across. So for drug development, we have to come together to do something very specifically significant and in a collaborative fashion. And as I was preparing for this talk, especially, I also found a very interesting article. It was published just last year. And surprisingly, this article is published by authors outside India. All 3 authors are from Australia and Greece. I was so surprised that they are talking about Ayurvedic formulations and potential COVID-19 therapeutics. Look at the irony. We as a country have this strongest base of Ayurveda in hand. Based on my understanding, we as a scientific community have very good understanding of phytopharmaceuticals, but why the people outside India who may not necessarily have a significant knowledge and understanding of natural products, they are writing authoritative papers and publishing it? Why not you and me? And we are not, why we are lagging into just putting right things in the right perspective on international platforms? So this paper in short spoke about how several preparations from Ayurveda origin and their phytoconstituents were found to be clinically beneficial to the patients who were suffering from COVID-19, and it acts as a preventive or diagnostic, preventive or treatment purposes. Why I am talking about this? And this next slide talks about several receptors of the COVID-19, especially spike protein, S2 receptor protein, and M-pro protease. And you can see on the square boxes and next to each receptor, you can see so many phytoconstituents like curcumin and piperine and whatnot. They have been found to be effective in mitigating or interacting with these receptors to be potential interventions for treatment of COVID-19. And we all are aware that why India's total death rate across the world was lowest as far as COVID deaths are concerned. I know there would be some gray areas where reporting may be or may not have been the way it should have been, but each one of us took some sort of or some form of natural interventions or Ayurvedic interventions or phytopharmaceuticals during the course of pandemic when it was in its peak. Meaning these interventions which we took had some way the other way impact on the clinical benefit to the patient in overall well-being and recovery from those symptoms. The reason why I am saying so is that we have an enormous wealth available, but I strongly believe, don't take me wrong, there is no concentrated and focused effort in a very specific direction, focusing one pathway, one drug, one group, one disease. That's not happening. Everybody is busy teaching their students for their master's or PhD thesis And again, this is you guys can correct me as a scientific team, scientific fellow, because I have studied in India and I know how system works. There are so many resource limiting systems there, but one student finishes his or her master's or PhD thesis, that topic gets closed then and there itself, period. You published one paper, submitted one thesis, the story ends there. Why we are not taking those very promising interventions or drug discovery programs which some of your students or your ideas have taken place? Why are not they going to the IND stage? Why we are not doing very structured, systematic, non-clinical safety and toxicity assessment of those programs? I am so concerned. Last week I was speaking with somebody in India. My one of my friend works in a pharmacy school. I asked him what kind of work you are doing. He said, "Sir, we are extracting some natural products." So my question to him was, if you are extracting some natural products, give me 20 examples of very promising programs which had been very systematically standardized, characterized. You have done several clinical studies and the drugs are available globally with appropriate global regulations. Forget about India. I'm not thinking about only India as a country. Why we have so much of manpower, exceptional talent, resources are there. Why we are not putting all these energies in a very focused manner? And that's something which I want to urge to all of us, that let us work together to do some meaningful drug development. I know there are limitations. Don't think that I don't know what are the limitations in terms of funding, resources, coordination, and expertise. There are several limitations, but top of that we have to build capabilities. But again, that's another context of discussions which could be taken as we continue our interactions. And as far as the global scenario about phytopharmaceuticals is concerned, US FDA has a guidance called as botanical drug development guidance. In Europe, they have their own system of classifying the drugs as a traditional use, well established use, or standalone use. In India, as you all are aware, ISHA is the leading body which is taking care of or leading the research and development in the space of Ayurvedic, Siddha and Unani medicine. But as far as the phytopharmaceuticals are concerned, CDSCO is the leading authority. Why I'm bringing this to your context? Because they treat phytopharmaceuticals at par with new chemical entities, and therefore we as a scientific community has a bigger onus to do a structured research which would have a clinical benefit to the patient. And this is again a tabular representation that what are the comparisons of phytopharmaceuticals drug regulations across these three important territories like US, Europe, and India. The best part, you can see that at least in India, we have drug regulation, phytopharmaceutical drug regulation is available. CDSCO is leading that, and there are several recommendations are already in place. Meaning the Indian scientific community along with the regulatory agencies are putting right efforts to streamline this process of drug development with a modern approach, because Ayurvedic drugs are one of the finest interventions, but unless we as a scientific community don't characterize it, standardize it, the global community is not so keen to accept those interventions, be those very effective. So we have a more onus, and I believe appropriate authorities are trying to put efforts in that direction. And you are an expert in this space of phytopharmaceuticals. I believe the participants who have joined are from pharmaceutics or pharmacognosy background, wherein you are quite aware that development of phytopharmaceutical drug is not and so easy process, meaning, when it comes from the natural resources, you have your own challenges, source of that raw material, consistency, reproducibility, and appropriate standardization and characterization become too complex, because the the extracts which come from herbal or natural origin have countless number of ingredients, and to identify which single ingredient is to be potent and effective, it becomes its own challenge. While doing so, as I said, therefore, if we integrate the several departments from the same institute across pharmacognosy, pharmaceutics, pharmacology, and chemistry, including analysis, if we all come together, work on one single program, my mandate, and if you would ask me how each institute, each pharmacy school in the country or in Maharashtra should work, each college should take one disease area, one set of molecules, and just take it to the clinical stage. Give it to a pharmaceutical company to develop it at a large scale. Then believe me, how efficient drug development would happen. And that gap of 10,000 diseases versus only 5% drugs available would be minimized in a pretty faster way. So I want to walk you through this interesting example where, now if you could recollect, I started with giving you some basics of drug development. What are the key components we should be talking about? Safety, efficacy, benefit risk ratio. Then I walked you through the landscape of phytopharmaceuticals, their strengths and weaknesses, or current challenges we are facing. So I just want to connect you to two classical examples where how the drug development should move on, and what are the key factors and from where which information comes in. So to give an example, dabigatran etexylate is a anticoagulant drug. Each one of us know warfarin. So this is the next generation anticoagulant, to make it simplified. So this drug is currently indicated for these several indications, especially to prevent the risk of stroke and systemic embolism. This drug is available as a capsule in 3 dose strengths: 75 milligram, 110, and 150 milligram. So now look at this lower box. It says warnings and precautions. So on the next slide, I will walk you through. This drug carries a black box warning, meaning US FDA, when it approves the drugs, if there is some safety precaution, the drug, again, the benefit risk ratio. So this is the risk component of that thing which I have been talking for long. Can you excuse me? Let me go and get some water. Just be online. Give it 10 seconds. So, as I said, each drug has both safety and efficacy evaluations. So this drug, called as dabigatran, has a black box warning, meaning that the drug has more safety concerns. If, as far as this drug is concerned, if this drug has an ability to cause thrombotic events as well as spinal hematoma. So these are the serious conditions if not taken with utmost precaution and under appropriate clinical guidance. Why this is important, and from where this information comes, right? It has got a black box warning. So how did it come? How do we arrive at this conclusion that this drug has a black box warning? Because in the drug development process, as I told you in the beginning, in the non-clinical toxicology evaluations, you as a scientist will always identify whether the drug has any issues as far as the carcinogenicity, mutagenicity, impairment of fertility, and beyond that, any safety signals. Because when you will do your dose ranging studies, short-term toxicity, long-term toxicity studies, you would be able to identify the potential shortcomings or safety signals in your non-clinical evaluations. Because what happens in a typical academic setting, what we generally do studies, or what kind of steps based on my understanding, right? Correct me if I'm wrong. You will identify a drug from natural origin, you will extract it, you and your team will run one in vitro study, one efficacy study in appropriate animal model, period. Most of the time, the research stops here. We generally don't go for identifying the mechanism of action of the drug, whereas that is critically important. Then we are more inclined towards assessing the efficacy, but not very rarely about the pharmacokinetic characteristics of the drug. So PK and PD are critically important part of any drug development program, be it phytopharmaceutical or small molecule. So these are very important things. So based on the non-clinical evaluations, you will run, as I said, once you open an IND, you will start a first in human study. So when you are in the process of starting the first in human study, the dose for first in human study comes back from, where does it come from? From your non-clinical evaluations, where you have identified NOAEL dose of your drug in appropriate species, rodent, non-rodent, and you will extrapolate that dose to the human equivalent dose, and you will give the lowest possible dose to first in human study, where you will assess the safety and tolerability of the drug. So, these are interconnected things always. So sometimes we ignore the experiment which we are doing in the lab, how it will impact to the bigger picture and the end user. Because this IC50 is not appropriately potent, your drug is not going, your dose of the drug would be higher. If the dose of the drug is higher, and again, there are other factors you need to pay attention to, is biopharmaceutics properties of the drug. So, it is, integration of multiple expertises, and therefore I sometimes feel very proud that I am a pharmacy student, because we have been taught everything what I am currently using it for my drug development programs and strategies. So if we can teach as a scientist or faculty to our student very good science and connecting the dots, I believe we would be able to produce a very efficient scientist who can in true sense will develop drugs going forward. So as I said, mechanism matters. Unless you don't know the exact mechanism of the drug, it becomes difficult to assess the efficacy and the safety component as well. Because most of the drug, to give an example, most of the anti-cancer drug have toxicity. Why does it have toxicity? Because it goes and binds to off-target binding. We call it as off-target binding, where the drug, also once in the body, it goes to every part of the body. So therefore, we have to have a clear-cut idea what is the exact mechanism of action. If you know it, you can direct the drug in a such way that it can be more efficacious as lower dose and with a higher safety margin. And therefore, we as a scientist should be able to connect these different moving parts as we continue the drug development. And as I said, once you have crossed that non-clinical phase of drug development, clinical pharmacology becomes paramount. And as I was explaining to you, there is not one study. We have to do series of studies as you continue the drug development program. You have to do first in human study, SAD, means single ascending dose, and multiple ascending dose PK studies. You study it in the healthy participants, then you go to the patient, and so forth and so on. I mean to say, you need to be doing so much of integrated team work as your programs move forward. I just now I mentioned biopharmaceutics. What is the bioavailability? At the end of the day, if your drug is not bioavailable, it has no meaning. What does that? What I imply from that? If your drug is orally effective or absorbable and it has bioavailability 1% or less than 1%, it becomes challenging. That means you will have to give a higher dose. Or to give an example, BCS class 4 drugs are neither soluble nor permeable. To give you another example, one classical example, a drug called as amphotericin B. It has a high molecular weight. It is neither soluble nor permeable. So it has to be only given by intravenous route of administration. So it becomes very difficult to give it orally. So, and it's a wonder drug, but it has its own challenges. So when we are getting into drug development program, we should be able to connect the dots. Just let us not work in an assumption that my job is to just run an experiment, say in vitro experiment. But let us try to connect the dots that this one experiment with appropriate IC50 value, if not done efficiently, it would impact my final stage of drug development. If I do that first experiment incorrectly, it has effect on the end user, which is my patient. So it has to be done very meticulously, very appropriately, and in a structured, rational thinking way. Now, I gave you this example of a modern drug, which is dabigatran. It's an anticoagulant drug. And as I was preparing for this presentation, I also saw that, how many drugs are approved in US. So I could find in US, they call it as a botanicals. There are only 2 drugs are approved under botanicals guidance in US. One is this cyanocatechins ointment, which is 15% for topical dermatological use, and this another drug called as Mytesi. These are delayed release tablets which are used for treatment of diarrhea. Now, look at this label. This is the prescribing information of one of these products, which is Vergin. It is a 15% ointment. I will just walk you through only the highlighted area and want to explain it to you how we as a scientific community can in true sense develop drugs with the modern approach. When I say modern, means not fancy approach. I mean to say modern drug development approach. So this Vergin is a topical ointment which contains 15% of cyanocatins. Is indicated for the treatment of external genital and perianal warts, right? This drug is applied 3 times per day to all the external genital and perianal warts. And this is contraindicated, or rather it is advised that not to use it for ophthalmic, oral, or any other use indicated. Why I am reading it for you? Because this is how, as each experiment which you contribute in your lab boils down to a simple 20-page prescribing information, which is the outcome of 10+ years of effort and spending of $2 to $3 billion. It compresses into just a 20-page document, and each line and each word in your prescribing information of an approved drug is worth millions of dollars, believe me. So every line and word written here has a significant impact on the clinical outcome in the patient. So now this drug has caused adverse reactions. This drug causes local skin and application site reactions, which is incidence is more than 20%. Can you imagine if we apply to 100%, 20% of the patient would have erythema, pruritus, burning sensation, pain and discomfort. Again, go back to what I said, benefit risk ratio. This is the risk where adverse reaction is nothing but safety concern. But at the same time, the FDA approved it because it would have a better safety profile, or not safety profile, I mean to say efficacy. Efficacy would be appropriate so that it outweighed the risk associated. Now, look at this. I also spoke to you about that modern drug, about mechanism of action. Dabigatran was a direct thrombin inhibitor. Now, look at this drug. The mode of action of Vergin involved in the clearance of genital and perianal warts is unknown. This drug is approved in 2006. Still, why it is approved despite not knowing the mechanism of action? Read the next sentence. In vitro cyanocatechins had antioxidative activity. The clinical significance of this finding is unknown. Look at the story. Pharmacodynamics of this drug is unknown. See, there are so many unknowns, but still the drug is approved. What I'm trying to say here, I'm not saying that it is just approved, because as far as the botanical or phytopharmaceuticals is concerned, the regulatory agencies are relatively, I'm saying relatively flexible to accept the NDAs which are submitted. Because this drug, what is the origin of this drug? You know, this drug has come from green tree leaves. So we, and especially in the Asian countries, we drink green tea very often. And in India, although we drink another tree, not green tree, but the context here is that this drug has a natural origin. They have identified, extracted the appropriate bioactives which are found to be clinically effective. And they did everything in a structured and systematic way. They did pharmacokinetic studies. See, this is again important thing. Pharmacokinetics of topically applied drug has not been sufficiently characterized as at this time. Still the drug is approved.
Very good.
Yeah. So, you know, and then the sponsor has conducted all the necessary non-clinical toxicology studies in terms of carcinogenesis, mutagenesis, and impairment of fertility. And after this, the sponsor also conducted 2 randomized controlled clinical studies where they found that the group of patients, almost 400 patients who were treated with 15% of this topical gel versus vehicle, had a success rate of complete clearance of the disease or clinical sign and symptoms almost in 53% of the patients versus 35% in the placebo group. What I'm trying to show to you here is that if we do a structured and systematic evaluation of your phytopharmaceuticals, the probability of going from one stage to the next step is higher. And again, the success rate of being it is approved and being useful in the patient is very important. So this is another example which I wanted to bring it to your notice. So to conclude, I am on the conclusion part of the discussion now. Whenever you are trying to develop a drug, you know, the best way is to interact with the regulatory authorities locally. If you are developing it for Indian market, we must go to DCGI sooner the better. Have a pre-IND meeting interactions where you bring your product and ideation and the drug development program to the regulator's attention. Discuss with them your current data which you have generated. Discuss the prospective plan. Okay, we have done efficacy study. We want to do PK study, short-term and long-term toxicity studies, and we call it as the IND enabling studies. So discuss every possible thing which you have concerns about across CMC, non-clinical and potential clinical development strategy. Once you have achieved that, because that also gives you as a sponsor or scientist to interact with the regulators and get them in line with your thinking, because it would be inappropriate to go ahead and spend a lot of money, time, resources and go to the agency and they will tell you, no, this is not the way the study should have been done. The clinical endpoint or endpoint in your animal study should have been entirely different. We do not accept it. Then we get demoralized saying that, oh, the regulators are not supportive. So we as a sponsor have, or scientists have to be proactively interacting with the regulators to be able to bring them on board, because they are not our critics. They are constructive critics. Why I am saying so? When I go to US FDA, we discuss everything very openly, very in a scientific way. There is no corner cutting. We do not say that, oh, this data has not come meaningful, let us not discuss with the agency. No, you as a scientist, you must be very open to discuss the shortcomings of your program also. Because if you hide it at this point of time, at the end, somehow it goes to the next stage, it will fail at a later stage in human study, because toxicity will be seen vividly. In animals, they can't talk to you, right? But humans will talk and they will complain about the toxicity or safety concern. So be proactive and open to interact with the regulators. And as you continue your drug development program, once you open an IND, you have an ability to use that intervention in humans. And I explained to you the list of studies in the previous slide. Then as you continue your drug development program, say there are different meetings to be discussed with the FDA. I'm talking more on the FDA side, that end of phase 1, you finished your first in human study. Now you are planning for your proof of concept efficacy study in patients. See, healthy participant is one ball game where you will just give it to me if I qualify the inclusion criteria. But I do not have that disease in the consideration, like say COVID. So we have to give that drug to the patients who have COVID with certain inclusion and exclusion criteria. There you will see whether the drug has been found to be effective. If so, at what dose? So you will have to do dose ranging study. You will have to do a lot of clinical pharmacology, drug-drug interaction studies. The effect of food means, because some drugs have to be taken with food and without food. So why so? So that's why it needs multiple experiences and expertise around the whole spectrum to be able to develop efficient and safe and efficacious drug. So once you have crossed these hurdles and you are taking the regulators with you in confidence, not to make friendship with them, but have a constructive scientific dialogue with the regulator, which has a meaningful outcome, you as a scientist and sponsor. That is why the interactions with the regulator are critically important. So, and if you have done it in the right way, I don't see anybody has to worry about the better outcome of your drug development program. So few quick slides, as you are planning for the drug development, you must be able to think about the target product profile of the drug. As I told you, what is the target population, target dose, target dosage form, duration of treatment, what could be the safety, efficacy, and all other things needs to be thought through in the beginning itself. So my recommendation that I urge all the faculty members to form smaller groups within your own organizations and sit and discuss, take views. If you are a phytopharmaceutical expert or pharmacognosy researcher, you must talk with a pharmacologist, because whatever the dose, he would, suppose you have developed a new herbal extract and which you believe you want to give it to appropriate animal model. But if you do, unless you don't talk with your pharmacologist colleague, it is redundant to keep on working in silos, and then when you go to him or her and say that my drug is not soluble, it is redundant effort. Or the dose is too high, which he or she cannot give by appropriate oral route of administration or intranasal route of administration. So there has to be dialogue, cross-departmental dialogue must exist. Then CMC, as I said, then we have to bring in your formulation scientist, pharmaceutics guy. He or she must be involved from the beginning. If we are not doing that, we are missing very important part. As I said, if your drug is not soluble, not permeable, what are you going to do? There is only one route of administration, which is IV. So now, can that become an easy dosage form? No, because for that you need an expert, skilled person to give any drug by intravenous route of administration. So what I am trying to say here, the whole team in a pharmacy school should work on one single program with a short and long-term goal. Believe me, that will bring better outcomes. And CDP, which is clinical development plan. And each pharmacy school, if we have good intentions to do right drug development, that pharmacy school must be connected to a hospital setting, and you must speak with your some of the finest clinicians in your town. Talk to them, discuss with them. This is the drug we are trying to develop. This is our intended route of administration. What are your thoughts and what are your recommendations? So unless that is not done in a holistic way, I don't see we are doing rational drug development or a drug development to benefit the patient, not benefit in terms of publication or filing a patent or submitting one thesis. If that is the short-term goal, that's fine, but we must have a long-term robust plan in place. And therefore, I believe we all have to work as a, we should be able to create and sustain dynamic and innovative. Read each word what I'm saying. Sustain, just creating a team which works for 3 months, 1 year, and then they scatter. No, it has to be sustainable, dynamic, and innovative, multidisciplinary. That's what I said. All the department in the same pharmacy school must talk and work on one single mission, one disease, one program. That would lead to develop new drugs. And then only, and considering India as such a highly populous country with resource limited setting, we all have to work together to develop drugs which are affordable and accessible to the poorest of the poor. So that should be the burning desire within us to work with that mission. Only then we as a scientist, believe me, all of us are among very lucky people on this planet. I'm saying planet, among 8 billion people, we are very select people blessed by nature to be able to think very differently. And the onus is on us as scientific community to develop something meaningful and which can be used for humanity. Otherwise, your PhD, my PhD is of no use unless if we are not doing something constructive. So to conclude, what I think, drug development is a teamwork, period. There is no ambiguity on that. And if we are working in silos, we are in a different world. And means the only short-term goals would be achieved by doing so. So therefore, and while doing so, while the team is efficient, what I call it, we should have a very high performing team available in each organization, in each pharmacy school, which has masters and PhD programs. They must be able to be working as a high performing team. And as I said, while doing so, appropriate regulatory interactions with the regulatory authorities are critically important. If your intent is to develop a product, intent is to be decided by individual one of you, or that organization have to set the target to develop what they want to achieve as a scientific accomplishment. And as I said, discovery to recovery and back to basics is the key, bench to bedside, and going from the bedside, finding out the limitations and improvising on those would in true sense will be a good strategy to continue drug development. And just to let you know, several years ago, me, Dr. Nilima Kshirosagar, and my colleague and wife, Dr. Priyanka, we had written this book almost 10 years ago, I would say, focusing on phytopharmaceuticals. So, if you want to read it. Again, no obligation. Just telling you that we as a team have to work very efficiently. And I would like to conclude with a quote by Sadhguru. If you shift from unwillingness to willingness, and from inertia to effervescence, your life will be joyful and effortless. So what I am trying to say here, that let us be willing to do something for the humanity. And if that shift has happened, I am so confident only good things will happen, and we should be able to contribute to the human health. And I think I will stop here. Happy to answer the questions and any comments your team has.
Yes, sir. Thank you, sir. It was really a wonderful session. Only one question in our chat box is there, sir. Our participant, one of the participant, Kapil Agarwal, want general talk on research perspective, sir.
Sorry, he want general talk on
research perspective, sir.
Means I'm not getting his question. Means you want me to talk about research perspective, or can you ask him or her to talk to me directly so
Yes.
that I can answer him or her?
Yes. Please, Mr. Kapil Agarwal, could you please unmute yourself and ask your question?
I think Dr. Kapil
Yeah.
is.
Meanwhile, Manoj,
Yes, please.
can I share few points?
Of course.
Yeah, it is always nice to listen. And what we heard last time, Dr. Narayana, you have continued, and we could definitely have connected the dots as per your title very nicely by elucidating with a lucid presentation. Even at the midnight of the hour, I think you have crossed 12:30 or so. So that way, even very nicely, and I am sure your message and your articulation of talking about phytopharmaceuticals and botanicals, we need to see how best we can efficiently work a country like India, while others are talking about botanicals, US FDA, why not India, which is the storehouse of Ayurvedic drugs? And in the times to come, I am sure the Mother India will come up to the level of thinking the way you have projected all our youth, particularly the scientists, those who are working at the earlier stage, they will take it a message, and years to come, we should see the light of the day with the botanicals touching the market. Good luck, all the best. And Isha, I'm sure you will be doing wonderful service ahead of time, and we will be together.
Thank you. Thank you. If there are any questions, concerns, I'm happy to talk to the participants, because I am not able to see them because we are talking virtually. But if they have any concern, questions, comment, critic, happy to hear. There are
Yes.
restrictions to industry to club with academia institution. Yeah, of course. That's why I want the industry and academia should talk to each other more often. And before that, my recommendation is let us start talking within the organization, because the pharmaceutics guy thinks he or she is the best in the world, whereas the pharmacologist thinks, oh my God, I don't need to talk to my neighbor in the next lab. That should not be the way I'm thinking. We have to keep our egos and personal, aspirations aside as we are trying to develop the drug. If we are so keen to develop, keep everything aside, focus on that program, look at the patient. If you really are concerned of doing drug development, go to a hospital or emergency care room, look at the patients there, and then come back and see that should I be busy discussing low energy discussions with my fellow colleague, or focus on finding solutions for disease in hand. So yes, and funding is a problem. It's a global problem, my dear fellow colleagues. But we have to come up with writing grants after grants, proposals after proposals. Reason why I am saying so, in US, average age of getting an RO1 grant, which is a premier grant where you can get up to $1 to $5 million, is 42 years of age. Average age is 42 years. By that time, that professor has applied for that funding several times. So if you are so determined to do something good, you will not leave that path and focus. So, but again, I have studied in India, I have worked in India. The primary focus of most of the young faculty is that my job is to teach one subject. I will go and take 3 lectures, 1 practical, boom, come back home, enjoy life. That's a different scenario. See, that's not a general statement, but what I'm trying to say, if we are really passionate to do right contribution to the science, we have to have that burning desire to do something unique and away from the conventional. Only then we will contribute and phytopharmaceuticals will come forward. Otherwise, one paper, one publication, one patent, finish. You will retire, I will retire in next 20 years. Then we will say, oh, kuch nahi hota hai. Sab kuch hota hai. That depends on your intent, and desire and energy where you want to focus on.
Yes, sir. Our next question is, many times herbal drugs are not dissolved in CMC as well as water. So what will be the solution? What need to be considered with this regard?
Good. So in that case, see, I don't have direct specific answer. That's why I said, if you are a pharmacognosy guy, you are facing this challenge, you should speak to your analysis guy or pharmacology guy. Unless you don't talk to each other or talk to your chemistry guy, ask him or her what is the solubility pattern or how we can modify the structure of your phytoconstituents to have a better solubility profile. How we can add another co-solvent to improve the solubility of that drug. So, that's why I am saying, don't hesitate to talk to your neighbor from different expertise so that you will be able to address this situation. Sorry, I don't have direct answer to your question that how we can change the solvent. It has to be very product specific, and I'm sorry, I cannot answer your question to that specificity.
Okay, sir. Our next question is, there are restrictions to industry to club with academic institutions. Then how make a bridge between academic industry for research activities?
For that, your organization's leadership should be the responsible entity. Your principal, your management, and the team of all the scientists or faculty at the institute should be so motivated to convince your leadership to go and bring in industry partners. And again, working with industry is not easy because their expectations are very specific because they are aligned with the regulators. And we as a scientist, we are most of the time in the exploratory mode. And we are not so systematic and so stringent at times. I'm not saying every time, but at times the expectations of industry is very high from rigorous reproducibility, systematic approach. That is possible what you said. Therefore, you have to convince your leadership. How you can convince it? First, prove within your group of faculty that you write a very nice proposal, get it funded, and then why not your leadership will not support you to go out, bring in industry collaborations. Again, industry will come to you when you have something very novel in your hand. You are working on some unique programs, only then industry will come to you. Industry will not come to you for a redundant, repeated, similar kind of research which is being more rampantly followed at many places.
Thank you, sir. Our next question is, sir, can you please elaborate some funding agencies in countries other than the India for phytopharmaceutical research?
Right to NIH. See, my recommendation is, India has so much of money. Why not you write some good proposals which are supported by good preliminary data? DBT, DST, CSIR and whatnot, we have lots of money. ISHA has tons of money. But that's why I said, if you are writing a very good, authentic, novel research proposal, why wouldn't these organizations support you? NIH and their standards are very high. I'm not saying you cannot get it. What I am saying, they would ask the same thing what I'm asking you. Do you have preliminary evidence which is reproducible? If the answer is yes, you don't need to go out of India. You have enough money in the country. But if you want to come to US, there are different programs at NIH and even US FDA also support research grants, but your program should be so unique and so novel that the competition is, as I said, 42 years to get an R1 grant. So do we have that much patience to be working in the same direction? So my recommendation, work with your colleagues, create reproducible preliminary data, write specific objective, objective 1, primary objective, secondary objective supported by your preliminary data, and then plan for the next studies. Let us not write, we want to do preclinical studies. What studies you want to do? Narrate the story, write down the abstract, write down the synopsis. You have to think through from now your concept to the end. Only then the funding agencies will fund your program. If you guys need some help in that, I'm more than happy to help you to review your proposals as a fellow Indians, and to boost your moral. I can devote some time to review your proposals, but those have to be critically, high quality only then.
Yes.
Otherwise, don't send me your students' PhD thesis synopsis, which you want to submit to the respective universities.
Thank you, sir. Next question is, established phytoconstituent categories such as flavonoids, terpenoids are not in clinical use. What will be the major limitations?
Wow. See, where again a difficult question to answer. My recommendation: flavonoids and terpenoids can be used in clinical practice, but how many have really taken it so seriously to develop in a more modern, systematic way? If that is the case, there is no inhibition of we having those into the clinic. But as I walked you to the example of the topical gel, which is approved as a botanical, those sponsors must have spent years together and generated robust clinical and non-clinical evidence. So terpenoid is again such a big class. We have to identify one specific terpenoid which you believe is effective in a specific disease condition. What is its mechanism of action? What is the non-clinical evidence, and then clinical evidence? And as I explained to you, what is the safety benefit risk ratio? What is its safety? What is its efficacy? So it's a very broader, context we have to think through.
Thank you, sir. Next question is, are the products with extract of herbs are accepted in a global market, or we need to concentrate on a specific phytoconstituents?
The answer is yes to both. Herbal extracts are widely used globally, and in one of my slides I told you these herbal extracts and dietary supplements and nutraceuticals, this market is growing exponentially, and US is one of the highest consumers of those. Again, phytopharmaceuticals is a stiff path to walk through. Therefore, it's a human nature. We want to travel the road which is easiest. Phytopharmaceuticals is a tough path to follow. You need more resources, funding, and patience to develop those drugs, and expertise as well.
Thank you, Dr. Manoj, for a wonderful session on the phytopharmaceuticals, the regulatory aspects of it, and the connecting dots. So to just conclude this session, I'll say some few words. Dr. Manoj in his talk elaborated the need of research on many unexplored diseases, core understanding and focusing on one specific target disease, target molecule, identifying the specific causative agents and the causes, and then finding the appropriate drug, which has a significant role in the drug discovery. The tagline of him, that is discovery to recovery, and turning the observation into intervention, is really a need of ours, as numerous potential basic research has not reached to the community yet. Dr. Manoj made participants here to understand the need of connecting dots from regulatory perspectives. He also emphasized on the need of work on the infectious diseases, especially in this post-COVID era and the scenario of drug resistance. He also emphasized on the drug safety first and then efficacy, and need to evaluate the benefit risk ratio of any intervention before claiming it for any potential application. Dr. Manoj also emphasized on the need of the translation of the research and the teamwork in the drug discovery process. Dr. Manoj connected the participants with phytopharmaceuticals by focusing on the global herbal phytopharmaceutical market and realizing the irony that we Indian are not putting our ancient knowledge about Ayurveda and herbs in efficient way and need to do the concentrated efforts in this field. With the live examples of dabigatran and veregin, Dr. Manoj elaborated on the need of the structured evaluation of the phytopharmaceuticals, like identification of the exact mechanism of action and the non-clinical studies for the human dose identification, the PK-PD correlation, etc., very efficiently. He also elaborated on the need of meticulous, appropriate conduct of various preclinical studies and need of connecting the various experts in the field with their studies and with their expertise for a effective drug discovery with a modern approach. In his conclusion slide, Dr. Manoj briefly discussed about the conversion of IND to NDA and mentioning the proactiveness of the inventor to connect with a regulator for the effective intervention of the diseases. So bench to bedside and reverse is a key area of focus. This line will be always remembered by all the professionals working in the field of phytopharmaceuticals. So with this, I hand over to the Gauri for the further session.
Thank you, sir. It was really a wonderful session. Now, I request you to please accept our token of appreciation. Thank you, sir.
Thank you so much. And I really enjoyed talking to you guys, and thank you for having me here. I wish I could have seen all of you in person, but maybe next time whenever I'm in India, we'll try to meet together and discuss in person.
Yes, sure.
Thank you, Dr.
Thank you.
Manoj. It is
Thank
really
you,
wonderful
Manoj.
to watch.
Thank you.
Now,
And
I
just-
request all the participants to please put on their cameras.
Hey, good morning, everybody. Hello, how are you? Yeah, I mean, I see such a wonderful experts, and I could see almost 144 participants. Are they from within Maharashtra or across the country?
Across India. Across
India.
Wow. So I am so lucky that I could share some of the thoughts which I have been able to learn with such an elite team of professionals and professors. And I believe, if we could work together as a team, things are possible, very much possible. But while understanding the limitations in our academic settings, I am quite aware of those. But if we are determined, things are possible.
Yes, thank you all the participants. Thank you, sir.
Thank you,
Okay.
Manoj. Thank you so much.
Yes, thank you all. Yes,
So we
go ahead.
wish you a very good night to you, because now it will be sleeping time for you there.
1 o'clock and 10 minutes. So let me
Yes.
see if I can sleep tonight.
Okay.